I'll also tell you one thing. After watching this video, 40 plus questions you can easily crack or easily cover or easily solve, and you can definitely get 160 plus marks in these exams after watching this video. That I can bet with you. Hello everyone, welcome back to our YouTube channel AgriAddict. So we are just starting our PYQ series, and I am starting with the plant science for the ICAR-PG exam, and I'll be discussing in detail about the questions of genetics from the year 2024, and likewise we'll be also discussing for the plant pathology, microbiology, seed technology and all other questions that are related to the other subjects within this plant sciences. And this will be for 2024, 2023, 2022, 2021 and 2020. So these videos will be lot more beneficial and useful for all of you. So don't skip any of these videos. So if you have not yet analyzed the previous year question papers, then this video will definitely help you to analyze the paper and to know what is important, to know what to read and what to skip and how to prepare for these exams and to secure the good rank in this exam of ICAR-PG. So let's get started. So before we start, let me introduce myself again. I am Ari Prasad, a PhD student from the Indian Agriculture Research Institute, New Delhi, from the Department of Seeds and Technology. I have secured All India PG rank of 48 in my ICAR PG exam in the year 2020 and All India second rank in the ICAR PhD exam for the subject of Seeds and Technology, and also have cleared the IPS A4 mains, plus like I have cleared the ASRB in the subject of ASRB net in the subject of Seeds and Technology. So this is in brief about myself. Okay, so now let's start discussing the questions one by one. The first question is about now this starts with the assertion and reasoning. So here I want to tell you about this assertion and reasoning type, right? See, the assertion and reasoning type of questions are more frequent. That is, like out of 120 you will at least get 30 to 40 questions which are mainly the assertion and reasoning. So what is important in that? This is like time management. Now you know, like for ICAR-PG exam you have like 120 questions, right? And to solve this 120 questions you have 120 minutes. That means for every question you have only 1 minute, but these assertion and reasoning questions will at least take 1.5 to 2 minutes to solve. So if you are allocating like 1.5 to 2 minutes for every single assertion and reasoning type of questions, and which are like 30 to 40 in the paper, then most of your time will be like consumed. You will be having very less amount of time to attend the other questions, and you would be getting no time to check the questions that you have answered, which is most important. See, many times what happens, like while we are giving it in a computer based test, so the options we mark, they will not be marked as it is like once we say. See, these type of technical issues happens, so that's why it is very much necessary that at least for once we should be able to check the answer what we have put in the exam, right? So for that you have to maintain a time where at least in 120 minutes, out of 120 minutes, in 90 minutes you should be able to solve it, or if not possible in the 90, at least in the 100 minutes you have to complete the solving of 120 questions, and rest 20 minutes whatever is left, which you have left and marked those questions, and you have to thoroughly check the entire paper for what you have marked. So for that at least you need 10 minutes of your time. So that's the reason, like whenever the assertion type and reasoning type of questions are asked, you have to make sure that you do not commit or consume like 1.5 to 2 minutes for every assertion and reasoning type. You have to practice well that you attend these questions within a minute or less than the minute, right? So how you can practice it? It's not possible directly going to the exam. You have to give the mock test, like several mock tests, not the one mock test, several mock tests, and each one of the mock tests you have to attend very seriously, and you have to track like how you are doing this assertion and reasoning and how much time you are taking to solve this. Only after proper regular practice you will be able to make sure that you attend the assertion reasoning in a short period of time. See, I'll tell you again, this is a serious warning. See, if you do not like concentrate on this and you do not try to practice to attend the questions in time without attending the mock test, if you are trying to go to exam and write, definitely you will be suffering with the shortage of time. That's for sure. You can ask anyone who has given this exam in the previous year, including me. Everyone will say that they did not get sufficient time if they have not practiced well. If you have practiced well in the mock test, you have already like very thorough about the mindset, you are thorough about the environment, like how would it be, like how the different kinds of questions will be asked, only then you will be able to attend it within a minimum amount of time and solve it correctly. And solving correctly is also another important thing, right? So for this, the practice, like only mantra is like practice. You have to practice, practice, practice. Only if you practice well, then you will be able to attend the questions. Sometimes the paper will be very easy, but the difference in the rank will be the time management. The one who like manages the time well, attend the questions well in short period of time, revise it and checks multiple times. So those students will get better rank than who is just not like considering all these things and trying to solve each and every question, taking their own time. So this is like definitely affect your rank. So please make sure that to practice the questions. So reading is one aspect, revising the content that you have read is another aspect. But at the end of the day, if you don't practice them, then you will definitely lose some good marks. Okay. And I'll also tell you one thing. After watching this video, 40 plus questions you can easily crack or easily cover or easily solve, and you can definitely get 160 plus marks in these exams after watching this video, that I can bet with you. Okay, so let's start with the first question, and whatever I said, it is for assertion and reasoning and statement based questions mainly, and also for like arranging the sequence. So these will take a lot of time, so please make sure that you try to like practice more and more, like how you going to attend this, so that in the real exam you will allocate minimum amount of time but solve it correctly. So let's start with the first one. So here the assertion is saying the eukaryotic cells, the newly synthesized polypeptide chains are often modified. So here they are talking about the modification. So this topic is related to post translational modification, which is a part of the gene regulation. So if you remember like what I have discussed in our online master course, so I discussed about the gene regulation in case of eukaryotes, right? And in that we have discussed about translational, like post translational regulation, if you remember. So in this is the concept. So what the assertion is saying, it is saying that all eukaryotic cells, the newly synthesized polypeptide chains is often modified after translation. That is, the polypeptide which are produced, they are modified. So this is the assertion. And what is the reason? The reason says the post translation modifications such as phosphorylation, glycosylation and the cleavage may occur to the polypeptide chain to attain its functional form. Here both the statements, like if we look this as a statement, both of them seems right. There is no problem in that. But here we have to link the assertion with the reason. So now to link it, I will tell you one very good tip. See, many times it happens, like if you have attended the mock test, if you have attended the previous year papers, many a times we fail to connect between the assertion and the reason, right? If you get it, you get it. So now I'll tell you like how you can make some connection with the assertion and reasoning. For that, you have to notice the keywords. Here the keywords is the polypeptide chain and the modified after translation. Now, this is assertion, and the keywords in the reason would be like attending the functional form. So how it is the keyword, I'll tell you. Here they are saying it is often modified. Now we know, okay, the after translation the polypeptide is often modified, but we want to know like why it is modified. So this why is answered in the reason or not, that is what you have to see. So whether it is answered in the reason or not. Now see, the post translation modification just such as like phosphorylation, glycosylation and cleavage may occur to attain its functional form. So this modification is actually necessary to attain the functional form. That is how the assertion is explaining this reason, or reason is explaining the assertion. So according to this, the first one is right, that both A and R are right, R are correct, and the R is the correct explanation of A. So if I want to recall the concept, like in the post translational regulation in the eukaryotes, what are the different processes or the mechanisms that come into play? It is like phosphorylation, glycosylation, methylation, acetylation. So these are the things, and I have discussed each one of them in bit detail, which is required for your ICRPG exams. So if you are preparing for this exam, if you are a student of master course, you already know it. But if you are watching this video for the first time and you have not enrolled for any of our course, please make sure from the regulation part you go through all of these things once. That will be definitely useful for your exam. Okay, let's go for the next question. The next question is again the statement wise questions. Here it is two statements are given. The first statement says the transcription is a process in which like genes DNA sequence is transcribed to make a RNA molecule. So here the process is about the transcription, and they are talking about the DNA to RNA. Yes, of course, like the transcription is the one where the DNA gets transcribed, the information gets transcribed into the RNA. Okay, so the first statement is right. It's very simple. Second one, for the transcription, the RNA polymerase binds to the promoter region situated upstream. So the keywords, I'll discuss these keywords: promoter and the upstream. What are these? And the RNA polymerase, most importantly. So if you remember from our class, like what I've discussed, I told you about the operon. Let's take a simple operon, right? The simple operon, if we take, like, you know, like it has a promoter region, it has an operator region, and it has the structural genes region. Now, if you remember, like promoter region, what is it? This is the binding site. Binding site for what? The RNA polymerase. And what is operator? Operator is the binding site for the repressor, right? So this thing connects here where the RNA polymerase is a binding site. Like promoter has a binding site for the RNA polymerase. And the next keyword is upstream and downstream. What is upstream? I discussed in the class. See, the transcription where it starts, it is called as transcription start site. If you remember, it is called as transcription start site. See, whatever sequence that are available behind this transcription start site or before this transcription start site, they are generally called as the upstream elements or the upstream sequence. And whatever sequence or whatever regions or nucleotides that we find below or after the transcription start site, they are regarded as downstream element, right? So this is the thing. So based on this, based on our understanding, I also discussed few other important things here. So based on this, what is right? The first one is right. Both statement 1 and 2 are correct. Okay. Next one. So it is a direct question. Whenever you get some direct questions, you should make sure that you save time here. You try to save time here by solving it in 10 to 20 seconds or maximum 25 seconds. So that is how you can allocate maximum time for the statement wise questions and the assertion and reasoning type of questions. Now, if the individual does not show a trait, even though they have the appropriate genotype, the trait is said to exhibit. Okay, so this is the question. Here, what kind of question is asked? Mainly definition of different terms. So for your exam, if you understand what is important, you have to know the literal meaning of the different terms that you get in the genetics. See, those questions or those kind of things can be asked in this way, where that trying to just ask you what is the meaning of certain terms. So make sure, see, if you are doing your self study, make sure that you list out all the terms that you get from the genetics topic and try to revise it from time to time. And if you are a student of master course, like every time I've clearly explained you what terms you can get, what kind of questions you can that can be asked in the exams, right? So let's solve it. I'll be solving it in bit detail. I'll not be only like telling you the answer and talk related to the answer. I'll also try to talk about all the different options which are available here, so that once you watch this video, this should be like maximum revision series where most of the concept of genetics should be revised. So let's start with the options one by one. I will leave this expressivity and the penetrance for now and focus on the partial and codominance, because if you see the question, the partial and the codominants are not related to this question, right? So this is already gone. So now I'll discuss these two first. What is partial dominance and the incomplete dominance, or incomplete dominance, partial dominance, and the codominance, if you remember. The partial dominance, if we can take an example, if there is certain combination which is having capital A and small a. Now, this capital A, if it is like regarded as a dominant gene, dominant allele, and small a is a recessive allele, if this is the case, then this cannot completely mask the effect of small a. That means this dominant, if it is dominant, it cannot completely mask the other small a where the dominant action is not clearly into the play. Because of this reason, the phenotype, if the capital A is giving one phenotype like red and small a is giving colorless phenotype like let's say white, then the combination of two, here it will not be red because it cannot completely mask the effect of small a. It won't be like white because it is other one is recessive. It will be intermediate to that, it will be pink, for example. And the literal example for this is in case of snapdragon flower color. Snapdragon flower color, which is like red, pink and white. Okay, so this is regarding the partial dominance, and you know it, I guess. And next comes to the codominance. So what is codominance? If again we take a combination of capital A and small a, or if we take a combination, then both capital A and small a are expressed. Both the alleles are independently expressed and they are present in the appropriate genotype. And one good example for this is like ABO blood group, and in which you can see the AB blood group, right? So this is the one good example to remember. So this is about the partial dominance and the codominance. Now let's talk about the actual concept which is related to the question. So what is it? It is about the penetrance and the expressivity. What is penetrance and what is expressivity? So let's start with the penetrance. So I will explain this with an example. Let's say there are 100 plants, and I'll talk with respect to one good example, which is like chlorophyll deficiency. Okay, now out of 100 plants, if I tell you that out of 100 plants, 80 plants have the chlorophyll deficiency and 20 do not have the chlorophyll deficiency. But the most important thing is the gene which is responsible for this, it is same in both the case. The genotype is same. The genotype, there is no difference in the genotype, but still only in the 80 of them it is getting expressed or it is getting visible, the symptoms are visible, and rest 20 do not have this trait or symptom. So then if this is the case, what we are seeing here, this is the phenomenon of penetrance, where it is like incomplete penetrance. Incomplete penetrance. If this is the case 1, let us take it as case 1, and if it is in the case 2, if out of 100 individuals or the 100 plants, if all of them are getting this deficiency, chlorophyll deficiency, then what do we call it as? It is like complete penetrance, CP, complete penetrance. So this is the concept of penetrance, where it is like, you can simply remember it as like presence or absence of a trait or a character. If the trait is expressed, if it is visible, if it is expressed in an individual, all the individuals, it is like yes. If not, no. Like if you take one individual, whether it is expressed or not, whether the individual has got that trait or not, that's it. Like it is simple yes or no mechanism. Okay. And when we talk of expressivity, again expressivity, just like the penetrance, has two different terms. In kind of penetrance, it was what? The complete penetrance and the incomplete penetrance. Similarly, in case of expressivity, I'll take the same example, like 100 individuals. Okay, now in this 100 individuals, when we talk about the expressivity, what if I say that not like 10 individuals have shown the complete chlorophyll deficiency and another 10 showed the deficiency only of the tip of the leaves, and the other 50 showed this expressivity at margins or at the center of the leaves, anywhere, like different variable. See, you see the variable type of expression which is not actually uniform. The kind of expression, all of them have the same genotype. See, they have same genotype. In like theoretical sense, all of them should express the same degree of expression. But we see that there is a change, like expression is in all of the individuals, no doubt, but there is a variable expressivity. So the term would be like variable expressivity in this kind of situation. And this is again the case 1, and if we take the case 2, there comes the other one which is called as uniform expressivity. So what is uniform expressivity, if you can tell me? So it is like all individuals are expressing the same chlorophyll deficiency pattern or the symptoms in them. That becomes the variable uniform expressivity. Now here just observe what they are trying to ask. If an individual does not show a trait, they are clearly saying that there is no trait which is being shown. So then it is the case of the incomplete penetrance. So this is the case of the incomplete penetrance. Hope this is clear, and I was also able to like recall the concept of the variable expressivity, the uniform expressivity, the incomplete penetrance, the complete penetrance, and the codominance and the partial dominance. And of course, like I'll be going slow and I'll be discussing each one of the questions. So if you are like already very thorough with these concepts, you can switch to like 1.5x and you can watch this video. And there is a lot of videos that is coming from our end. So we are also planning for like a PYQ series. We are planning for like revision series, like marathon series, weekly mock test discussions. So your feedback, your comments on the videos, like what kind of content you want, how you want it, how we should be delivering it. If there are any suggestions or feedback, we would be really happy to get it from you on the comment section. So please comment on this video, like if you have any specific queries, or if you love the content, also share some love so that we'll be motivated enough to make such videos. Okay. Next question, again it is about the assertion and reasoning. So given below are two statement. One is, okay, I won't be reading it. So assertion, what is it saying? During DNA replication, the lagging strand is replicated in short pieces. Okay. Assertion is saying that during the DNA replication, the lagging strand, if you remember the Y fork, they are saying that. So let me just draw the lagging strand first. So in this, which is the lagging strand? If you can answer. So this is the lagging strand. This part is the lagging strand, right? So now they are saying they are synthesized in the short piece. As you can see, the short pieces here. So then the assertion is true. Next, what does the reason is saying? The reason says that the DNA polymerase can synthesize the DNA only in one direction. Again, many times it happens, you cannot actually connect between the assertion and the reason. But I told you one very clever trick is to make sure that in the assertion you find the keywords and in the reason also you find the keywords and try to match them. First, the keywords here is it replicates in the short pieces in the lagging strand. Now, why it should synthesize in the lagging short pieces in the lagging strand? Why it can't be continuous? Whether it is explained in the reason or not, that's what we have to observe. So here it is saying that it can synthesize only in the 5' to 3' direction. That is why it is in the short pieces. Whether this is true or not, let's see. See, if in case the DNA polymerase is able to synthesize in both the direction, which is 5' to 3' and 3' to 5'. In this case, whether there is a chance of getting the short pieces, tell me yes or no. No, right? Because if it is like in both the direction, in the opposite direction, this synthesis can begin, and if it is beginning in this direction, there is no short pieces. It would be like continuous, right? As an unwinding goes, like it would be continuous in both the directions. This short pieces formation or the lagging strand formation, it is because of the fact that the DNA polymerase only capability of synthesizing the DNA in 5' to 3'. So this is how the assertion and the reason both are linked, and here the correct answer is A and R are correct, true, and R is the correct explanation of A. Okay, make sure to link the questions like this, which are assertion and reason, and try to answer there. Okay. Now, this question says it's about the formula directly from stats, and it's mostly from genetics also. So let's see like what it is saying. The formula is like sum of observed minus expected whole square divided by expected is also known as. So they have given some formula in a statement form, like we'll give it in the formula like O minus E, which is observed minus expected, like sigma sum ofs, and divided by expected, it is equal to what? So if you just look at this formula, what is this formula? You know it, right? It is the chi-square test formula. So it is very easy to say. Now I'll talk about the stats. If you are preparing for these exams and willing to crack the plant sciences, the genetics, plant pathology, plant breeding, microbiology, seeds and technology, these things only will not help you. You have to cover the other subjects like biochemistry, biotechnology, crop physiology, and the stats. After this, like these are the primary priority. After that, you have to also cover the general agriculture topics from the agronomy, soil science, extension, and few parts of economics. These are some of the very key important areas which you have to focus, and one among them is like stats. For every year paper, for every subject, stats questions are compulsory. Like one or the other questions will be asked from stats. So your foundation, like not too much advanced topics, like foundation of the statistics like concept should be very clear. Like how to get it clear, like we have uploaded like 6 different videos in our YouTube channel by making a separate playlist of statistics, in which like our mentor Saki have nicely explained each one of them in a very detailed way, and also very in concise way. Detail doesn't mean that it is very vast. In a very concise way, within 6 videos, almost all the things that is related to the statistics which is required for this ICAR exam is clear. So if you are not yet watched this video, the links will be available in the description box. So please go and watch the video and cover the statistics in just 6 classes. It is just like 6 hours, 6 to 7 hours. You can clear the complete statistics. And if you are a student of master course, you will be also getting the study material of it. Just go and access the study material once and read it from there. And if you get any doubts, clarify it from our mentors and get it sorted. And that's how like you will be prepared for the stats. Okay, now coming back, so stats is fine, the chi-square test is fine. What is the other name? Like it is also called as what? Goodness of fit test. And now what other things are important? See, whenever you encounter questions in the previous Q papers, getting the right answer is not the only thing, right? Other than that, you have to also get to know about the other options. Whenever you see the 4 different options, no matter what is the question, if you are well aware of all 4 options, right, you have some idea, you have some knowledge about all different options in the questions that you get or you encounter, then assume that you are prepared well for this exam. Here, if I simply randomly ask about what is F test, what is T test, if you are able to answer, then you are prepared nicely for this exam. Now, and also variance. So here in the stats, like mean, median, mode, variance, standard deviation, and different kind of tests like F test, Z test, T test, chi-square test. So these tests are very important. So you have to know about like what are these tests, where are they used, what are their application. Now if I have to just simplify this T test, why it is used, it is like to compare, for the comparison of 2 sample means, for 2 sample means. And if it is for F test, it is for more than 2 sample means. And the variance, you know it already, it shows about the measures about how much variability it is there for the central value, right? How much, what is the range of this? So that is what variance is. So for this, like please know the formula of all this test and its use case. So this becomes very much important. If you do not know about it, if you are not aware of it, please go and watch the statistics videos, from which all of these things will be very much clear. The next one. So which of the following is a term for the plant or the embryo that contains only the gametic sets? So here it is asking about the gametic sets of chromosomes. Now again you have 4 options, in which like 3 might be confusing again. What is aneuploid, what is haploid, and what is monouploid? So aneuploid, you know it already. If you know it, so what is it? Aneuploid, it is like chromosome, like variation in the chromosome, but only in the single set, single chromosome or a single set. If there is any change in a single set of chromosome or just a single chromosome, that is under aneuploidy. Okay, alteration to the chromosome. And what are the examples? The examples for the aneuploidy, you have trisomy, you have tetrasomy, you have monosomy. So all these are examples. And also you should be knowing the formula, like it is 2n plus 1, tetrasomy is 2n plus 2, and monosomy is 2n minus 1. And if I talk about nullisomy, it is 2n minus 2, right? And there is one more thing, like there is double monosomic. So what is the formula for double monosomics? It is 2n minus 1 minus 1. So this is about the aneuploidy. So this is clear. Now what about monoploid and haploid? So what is the difference between the monoploid and the haploid? See, if you are considering a diploid, then the monoploid and the haploid might be same. Both are same only. But if it is in the polyploid species, it is different. In the polyploid, both the monoploid and the haploid are different. So what is haploid? Consider or know that haploid is actually the gametic chromosome number. And this, will the gametic chromosome number will be always like a single set? Is it true for in all the cases? So single set of chromosomes is only true in case of diploid. If there is a diploid, then it's in its gametic stage, it will be having the single set of all chromosomes, right? This holds good. But what if I say that it is a polyploid? then it is in its gametic stage. Will it have only single set? No, it will not be having a single set here. So that is the main thing here. For the haploid, it is just the gametic chromosome number, which is exactly half as the total chromosomal complement. But what is monoploid? A monoploid clearly explains about the single set. No matter like what is the size of the genome or how many different sets of chromosomes are present, when we have single set of the complete chromosomes, that is denoted by the name or the term the monoploid. So that is the like explanation for all the four different terms. The fourth term I left it. What is gametophyte? It is a stage of a sexual production where the gametes are being produced, right? This is the gametophyte. Next question says, like, both plants and animal cells contains some small organelles called as peroxisomes, which are dedicated to the metabolism of substances. They're asking the role of peroxisomes again. So a lot of people ask, like, sir, what is important from the cell organelles? What we have to read? Like, we have read this cell organelles from the 11th and 12th and in the UG, and now also we are trying to read it. But what we have to read? From the cell organelles, if you do not know, you have to list all the cell organelles, its structure, you have to know about its structure, and third column should be its function. So you should be knowing about it, where it is located, and about its function, whether it is located in the animal cell or the plant cell, plus its structure if there are any specificity, plus the most important thing is its function. What are the functions of the different cell organelles? You have to make this list compulsorily, and you will definitely get some questions in the exams from the cell ultrastructure. Here they are talking about the peroxisome. So what do you like remember when you see the term peroxisome? So what do you remember? What do you remember? So I remember, like, I'll tell you like what I remember when I see this peroxisome. It is like beta oxidation. I remember beta oxidation whenever I see the peroxisome. And what is this like beta oxidation is related to? It is like breaking of fatty acids. It is the breaking of long chain fatty acids, breaking down of this. This is what the beta oxidation involves. So if that is the case, the fats are already involved. So in the answer, we already got the answer. It is fats and the amino acids. So along with the fats, these peroxisomes are also involved in like removing the amino group from the amino acids. Later, which this will again get mobilized or metabolized. So these are the two important functions. So if you want to know about the other important functions, I have clearly mentioned about what are peroxisomes and what are they involved in. Like, they are involved in like fatty acid metabolism, amino acid metabolism, and also they are involved in ROS detoxification. So these things also becomes very much important. Anyhow, like, I'll be sharing this PDF with each one of you, so you know it, like how to get this PDF. If you do not know it, so just message us on our WhatsApp number 8920321513. So we will share this PDF. Okay, in this PDF, what help you will get is, like, I have also tried to compile the information for the different options for few very important questions. Almost like half of the questions will have it some kind of explanation which will be helpful for your exam. So you can get the PDF by texting on this number and by just completing some simple few tasks. So you'll get this PDF. I also talked about gave the information about the other options. So let's talk about starch. See, what I'm trying to convey is, you see 4 options here. From the 4 options, you have to recall like what you know about those. Like, for example, if I ask you about fiber, tell me something about fiber. So you have to know certain things about fiber, at least basic things. Like, if I ask you about starch, you have to know certain things. For example, starch, where it is synthesized. See, starch has like 2 different types. Like, one type of starch, it is synthesized on the chloroplast. And what is this chloroplast? It is photosynthetic plastid. And the second, like, it is amyloplast, where it is synthesized. And it is non-photosynthetic plastid. And in the photosynthetic plastid, the chloroplast, so where the starch are synthesized, they are like collected or synthesized in the daytime, and you know, like, they are mobilized in the night time, right? And when I talk about the amyloplast, these are the non-photosynthetic plastids. In the non-photosynthetic plastids, and the starch are actually stored as like in the tubers. These are stored. So this is about the starch. Okay, and this same information, like, I have tried to give it here, like in the tubers, seeds, and the roots, this starch is stored, which is in the ameloblast. Okay, and what is fiber? Like if I ask you about the fiber, what is fiber? What you'll answer? The fiber is like part. It's mainly of, you have to remember, fiber. This is a part of plant cell wall, and it is not found in the animals, right? And fiber components, what it is made up of? It is made up of cellulose, hemicellulose, pectin, and the lignin. Okay, and what are these synthesis sites? Everything I have tried to compile here. So this might be of some use. You can expect some kind of questions. So please make sure you at least go through it once. Okay, and in the last table, I try to like make some table on which, like in a very simplified way, I've listed all the cell organelles which are available and in which of the organisms, whether it is both plant and animal, or it is only the plants or only the animals. So such kind of things are already clearly mentioned. So these are a very easy type of questions from which you can easily score the marks, and here you can't commit the mistake if you are wanting to get a good rank in the exam. So all this comes with again the practice. I'll tell you, please practice the mock test, don't ignore it or don't neglect it, and don't think that okay, I'll prepare till like last week of the exam, in the last 1 week I'll only attend the mock test. Don't do this mistake. If you are preparing, if you are just started, still try to attend one mock test every day. By the time you reach the end to the day of the exams, most of the things will be clear, and also you will be confident about attending the exam. Okay, next thing, it's about the Operon model. This is very easy to answer. Like who proposed the Operon model for the gene regulation in bacteria? It is like Jacob and Monod. They are trying to confuse you by giving the wrong names. So this is gone. But I told you, I'll be also discussing the other options. Now tell me, like what is the contribution of the Sanger, if you remember, and what is the contribution of Watson and Crick? So Sanger, the first person to deduce the DNA sequencing method, plus the protein, the first protein to be sequenced, which is it? It is insulin. So these are the two important contribution of Sanger. Fine. Next, Watson and Crick, you already know it. It's about the DNA double helix model. So this DNA double helix model, you also have to remember the year. It is 1953. And for this model, like how many got the Nobel Prize? So for this, the Nobel Prize was given to, like you know, it's Watson, you know, it's Crick, and third, it was also given to Maurice Wilkins. So why it was given to the Maurice Wilkins? So along with the Maurice Wilkins, there is Rosalind Franklin. Both of them worked with the extracrystallography, which helped them to get the image, wet image of DNA, using which the Watson and Crick have actually deduced the DNA double helix structure. For this reason, like it is awarded to these three scientists. But why not to the Rosalind Franklin, if you know it? So she was already dead when this was announced, the Nobel Prize was announced, and you should know that Nobel Prize will not be given postmortemously. Okay, so this is done. Next, so same thing I try to mention. So if you can remember also the years, remember I did not talk about the operand model. It was given in 1961 by Jacob and Monod. Okay, and it was in which? It was in the E. coli. Explain about the gene regulation in the prokaryotes. Okay, next again we come back to the assertion and reasoning type. Here the assertion says that during the meiosis, the genetic recombination occurs which leads to the genetic diversity among the offsprings. Okay, next the reason, what the reason is saying, the genetic recombination is facilitated by the crossing over of homologous chromosomes during the prophase 1 of meiosis. Here again you see, you might sometime fail to connect between the assertion and reasoning. So that way always occurs. That this is the third time I'm saying to you people. But again you have to just check the keyword and trying to connect with the keywords. In the assertion, let's see like whether it is correct or not statement. During meiosis, genetic combination occurs which leads to genetic diversity. This is true. This is because of the crossing over or the event of genetic recombination, we see the variability among the offsprings. So that's very true. Next, the genetic recombination is facilitated by the crossing over of homologous. Yes, crossing over occurs between the homologous chromosomes, and it is during the prophase 1. Yes, it is during the Packettine stage of the prophase 1 where this crossing over takes place. Now how to link it? So they are talking about the genetic diversity among the offsprings. So how like genetic variability or diversity is getting? It is because of the exchange of the physical segments. when the physical segments are exchanging, the genes, the sequence, they are also exchanging, because of which we are getting the genetic diversity. Is the reason linking with the assertion? Yes. So that's why the answer is like both A and R are correct, and the R is the correct explanation of A. Okay, next again the statement wise question. The statement 1, the DNA replication proceeds in a bidirectional manner from the multiple origin of replication in the prokaryotes. So they are saying that in the prokaryote cells there are multiple regions, like multiple regions for the start of replication, or multiple origins for replication. So this is for the prokaryotes which they are saying. And second one, the Okazaki fragments or the short DNA fragments synthesized on the leading strand during the replication. So we have already seen it, it is not on the leading strand, it is on the lagging strand. Okay, so this is false, the first statement. Now you have to remember, and if you have gone through my classes, you should be knowing that in the eukaryotes, this is where we call it as multi-repliconic, and in the prokaryotes it is just a single unirepliconic. That is, there is only one replication start site from the where the replication will start and end, and this is only one in case of prokaryotes, and it is multiple in case of eukaryotes. So according to this, so both statement 1 and statement 2 are false. Okay, next, in the case of lacoperon, see, you are already seeing the question of lacoperon twice. In the case of lacoperon, the presence of the glucose in a cell or a growing media will lead to, okay, so this is a simple concept related to the lacoperon, but they are not trying to ask you about the operon now. This, the one which we mostly study. Now if you remember about the gene regulation, I talked about 4 different types in case of prokaryotes. I told you about positive inducible system and negative inducible system, and I told you about positive repressible system and negative repressible system, if you remember. So in this, what are the examples for the negative inducible? Yes, lacoperon mediated by what? LAC-I gene or LAC repressor. This is the example. And for positive repressor, there is no example. For the negative repressor system, it is like tryptophanoperon, remember. And what about the positive inducible system? Here the example is CAMP mediated regulation. This is the example. Now they are asking, in case of lacoperon, the presence of glucose, this is related to this first one, which is positive inducible. See, according to the positive inducible system, when the glucose concentration is high in the cell, then there will be lower CAMP, and because the slower CAMP is present in the cell, which has actually the activity of going and binding the promoter region and making it accessible to RNA polymerase. Its primary role, the primary role of CAMP along with the CAP, which forms a complex, activated complex, CAMP with a CAP, catabolite activator protein. This should actually go and bind to the promoter region, and because of which it will make a conformational change in this region, which will allow the RNA polymerase to come and bind here, through which the transcription occurs. But because the presence of high glucose in the cell, so this activity is abolished and there would be no activation of transcription. Now this case, like what is saying, like this is called as catabolite repression, where the catabolite, which is to be broken down by the catabolism, it is repressed, where the lactose catabolism is repressed. That's what we see here, right? When the glucose is high, the CAMP cannot come and bind here, and overall the lacoperon will be switched off. So lacoperon will be switched off. What does it mean? It means like there will be no lactose metabolism, or there will be a lactose repression, which is the catabolite repression. The answer is catabolite repression. So here I have discussed about the positive inducible system, the negative inducible system, the positive repressible and the negative repressible system. And the examples, these 3 examples are very important. Please make sure to remember. For negative repressible, it is tryptophanoperon, and the negative inducible system, it is lacoperon mediated by LAC-I gene, and for the positive inducible, it is CAMP plus CAP mediated regulation. Okay. Next, Now we have got a question of match the following. See, I'll tell you, the match the following questions are very easy to answer, mostly very easy to answer. If they are asking you like 10 questions, at least 7 to 8 questions can be easily answered. If you know at least to match one or two, rest of will be matched automatically. So this is why I'm saying this is like, if you are wanting to become a topper, then you you should not make a mistake at any cost in this match the following. Everything should be very clear, and all the questions that come in the match the following, you should be answering it properly and correctly. Okay, now there are 4 different terms. I'll read it out: discontinuous eating, the destruction of all viable forms, ionization, and pyramid damage. As soon as I see the pyramid diamondization, I'll go with the UV. We know it already, like you know it. So next thing, the ionization of the biomolecules, biological molecules. There are 2 different radiations which are available in the option. So it was regarding the UV rays and the gamma. And we already know that U is a non-ionizing radiation. So which one is left now? The only gamma is left, which is obviously the ionizing radiation. So this is also matched. And next, the discontinuous eating. See, if I want to talk about the discontinuous eating, which is like actually the tyndallization. Tyndallization is a simple process where there is a discontinuous eating. For example, for a short period of time, they will go for eating, and for a long period of time, they will go for cooling or leaving it to allow to cool. And next, the for short period of time, they are again going for eating, then allowing it to cool, and then short period of time eating, and then allowing it to cool. And this cooling period is high, and this heating period is low, and overall this will help for killing the endosperm, which are resistant bacterial spores which are formed, the endosperms, bacterial endosperms. It will help to kill these endosperms. Okay, so this is regarding the 4 different options. Like it is like dimerization UV rays, ionization of the biological molecules by gamma rays, discontinuous eating by tyndallation. The one is left, which is destruction of all viable forms. It is like steam sterilization. This is very easy to understand, right? Okay. So I also given some information regarding the tyndallization. If you want to refer it, you can go and refer this. Okay. Okay. The next question. So question number 51. Now this is like arranging the following. The next question, this is about arranging the following. Like we have seen the questions like statement wise, assertion reasoning, and the direct questions, and also the match the following. Now the other type of pattern, the other type of questions that are asked is like arranging. Here also, like sometimes you might have to spend like more than a minute, but otherwise most of the time you can solve such questions in within a 1 minute. So let's see like what it is asking. See, arrange the following concepts or the principle in a chronological order from oldest to latest, right? In ascending order. So that's what they are saying, like from oldest to latest. So like a lot of students ask me this question, sir, like for every event, for everything that has been done in the past, like do we have to remember the years? So not directly, like they won't be asking you directly the years every time. If they ask you like to arrange the things chronologically on older to latest, that is again the same thing, that you have to remember the years. If they do not ask directly, but still like if you get such questions, if you do not know about the events and its years at what time they have taken place, then it would be like difficult to answer. But still, even if you do not know the exact year, just by applying some logic, so you'll be able to answer it. Now it is like gene for gene hypothesis, dominance hypothesis, transforming principle, and concept of heterosis. Now like I'll also try to discuss the other things here. Transferring principles, you know, like it was in 1928, right? It was by Frederick Griffith who gave this concept. And in which organism did he give? It was in Streptococcus pneumoniae, right? So this is the first thing. Next, concept of heterosis. The heterosis concept was like started very like early. There is no clear cut the event that has been described or it has been published or reported. But the concept of heterosis, like if we talk about the term, like who gave this term? The term was given by Schell, who was a mace breeder, and it was in 1914. Okay, so this is how you can remember it. But if you talk about the term, like this describing this concept of heterosis, it was starting from the hybrid vigor. Like there are a lot of other names which was used for the same concept, and started with the Charles Darwin, even much before the Charles Darwin also. So the exact year for the rise of this like concepts you might not be clear, and for in the different books you get the different year names. So do not worry about it. Just know that the exact term for the phenomena which was like heterosis, the term was given by Schell, and it was in 1940. And much before giving this term, like that coining the term the heterosis, there were the dominance and the another one is like over dominance hypothesis. What does this both related to? It is again related to explaining the genetic basis of heterosis. So it was actually explaining the genetic basis, like how do we get F1 which are superior over the parents. So that was explained by one is dominant hypothesis, other one is over dominant hypothesis, over dominance hypothesis, right? So here, if you remember dominance hypothesis, you all know it, it was given by Davenport, right? And it was also extended by some other scientists later. And if you remember the year, it is in the 1908. And in the same year, the over dominance hypothesis was given by two scientists who were again Shull and East. And this was also in 1908. But first came the dominance hypothesis and then came the over dominance hypothesis. And if you talk about gene for gene hypothesis, which was given by H. Floor, and it was in the year around 1940s. Exact year you don't get it anywhere, so do not worry about it, but it is mentioned that it is in the 1940s. Now based on this, try to arrange it. So of course, like it starts from C, 1928, sorry, not C. It starts with the dominance hypothesis, B, and next it goes with D, and next it goes with, sorry, B, it goes with C, okay, B, D, and goes with C and A. So this is the answer. B, dominance hypothesis, D is like over dominance hypothesis, C is like transformation principles, and the A is regarding the H. Floor, which is gene for gene hypothesis. Okay, so according to this, the answer will be B, D, C, and A. Okay, so this is the right answer. See, in all the options, like B is common for two options, C is common for two options. So that's where you might get confused. But if you know the year of the transforming principle and the dominance hypothesis, then immediately you can eliminate these two and you can look into these two. And if you know about the concept of heterosis and you know it is 1914 and you know that it is in 1940, then only like you will be able to choose the C over D, and the right answer would be D, C here. So it's very much important that whenever you read any concepts, any events, try to keep everything in one place. See, I see like a lot of people do not make their own notes. So that's completely up to you, like whether you want to read it in your own notes or you are already like well versed in reading the textbook directly. But when it comes to the revision, when it comes to the entrance exams, like where such questions will be asked for last minute revision, like you need some crisp note where you have all the things at one page so that you can revise it nicely. So I would recommend you, like even in the last 30 days, take the help of your friends, do some group discussions, like someone will have some material, you will be having the other material, just try to exchange it. Don't think it in a competitive way. If you are in a same place, just try to like enrich yourself with the knowledge that they have also discussed the things by this. So you will be able to get all the things at place and you will be able to revise it nicely. And also, like most importantly, like while you are preparing, having the motivation, having the right things in place is very much important. And how you get the motivation? If you have a good surrounding, if you have a sound environment, the positive environment, all your friends are preparing well, they are very up to date, they are very disciplined, they wake up early, sleep early, and they are maintaining certain schedule. So automatically like you will be also like engaging with them. This is how like you enjoy this journey. Basically, like this would be the last time where you can enjoy till ICRPG. So once you prepare for ICRPG, that will be the lengthiest time that you might have prepared for any exams. This is from my experience. Like I have prepared for like ICRPG exam for 1 year. That was the last exam that I was able to prepare for 1 year. After that, every exam is only for 2 months, only for 1 month. Even the SRF was only for 2 months. So just try to enjoy the phase and simultaneously try to get some good knowledge about the subject. Okay, and that all comes with a good environment. If you have, if you are reading alone in your complete universities, that is a different thing. So you have to be self motivated so that you do the things every day. Okay, the same thing, like I have tried to like give it in the table form. The hypothesis is done, dominance hypothesis is done, and transform principle is done, the concept of heterosis is done, and the O dominance hypothesis is done. There are two other concepts related to this heterosis and the hybrids, like which is like double cross hybrids. Who gave this double cross hybrids? Jones, and it was in the 1918. And if you talk about the top cross, you know it is Davis, and it was in the 1927. Similarly, like I want you to like have compiled things of the events which takes place in the genetics separately, which takes place in the plant pathology separately, microbiology separately, seed tech separately, and plant breeding separately. If you have like 4 different notes on the entire set of events that has been taking place, and you can revise it in just half an hour, 15 minutes of each of the subjects, and if you do it multiple times like every day, So most of the things will be very clear. Just try to follow it. So the next question, like it is one liner again. So the aneuploidy describes a numerical change in the part of a genome, usually from the dosage of the single chromosome. And if the chromosome or a segment is overrepresented, it is called as, they are asking like, if it is overrepresented, then what is it called as? The term, they are asking about the term. So there are 4 different terms here, right? For a few, like as soon as you see the question, you will get the answer for sure. But for many, what happens, like whenever you see such kind of questions, still you know the answer, you know the concept, but as soon as you see the options which are like more or less looking very similar, like you get confused. So in this case, there are high chances that you will commit the mistake. So how to avoid this mistake? Make sure like you just try to eliminate the options which are not with the in accordance with the question. So let's start like decoding the meaning of it. First, let's start with what is hyperploid. See, hyperploid is something having the extra chromosome or the chromosomal sets, the entire chromosomal sets or the single chromosome, right? And it can be again divided into two different types, the aneuploidy and the euploidy. And this euploidy, if you remember, it can be how many different types, like examples, like you can have like monoploids, you can have haploids, and you can have polyploids, right? And in this polyploids, again, what are the different types you have? Like if you remember, it is auto and it is allo. So what is the difference between auto and allo? The repeations which are present in this auto, they are of same genome. The extra chromosome sets are from same genome. But when you talk about the allo, it is like 2 or more distinct genome, right? So this is about the auto and allo. But what is euploid and what is aneuploid? So aneuploid in the first place, if you remember, it is like loss or gain of a single chromosome or a single chromosomal set. That's it. Okay. But euploids, where there is a change in the entire set of chromosomes. That is what the euploidy is. So in this, you can also divide it in such that like it can be like hyperploid and hypoploid. So what is the difference between the hyper and hypo? Like it is extra, it is like less, where there is a chromosome segment which is lost. Okay, and in the hypoploid, if you get a question like whether the aneuploidy actually belongs to the hyperploid or the hypoploid, so it can belongs to both, because whenever there is a addition, like for example in the trisomy, then we talk about trisomy, this is an example for what kind of condition? It is an example for hyperploid, right? And if it is like monosomic, then nullisomic, it is an example for hypoploid. And hypoploid, so how can define? Like it can be like loss of a chromosome of chromosomal sets, any number, simple, in the simple terms. Okay. And haploid, this is also clear. The polyploid, all this is clear. So now since they are talking about the overrepresentation, which is like adding extra, the answer to this question will be like hyperploid. Okay, simple and very clear. So I have added the details. So this is from our online master course. I have added the details in this slide so that you can revise it in one go. So just have a one glance at it. The next question. In the DNA molecule, if the 30% bases are adenine, what will be the percentage of cytosine? If you are well aware of the concept, you will directly answer this. But if you have not read it, or as soon as you see the question, you will think that okay, there is some formula, like there is some formula, I have to remember the formula, But just see the question clearly, it is a conceptual question, not a formula based question. So what they are saying, like in a DNA molecule, 30% of bases are adenine. They are saying like A is equal to 30%. They have given it in the percentage. So if you remember the feature of the DNA double helix, you know it already that according to the parity rule, A will be always equal to T and the G will be always equal to C. And if you talk about the number of bonds, there are 2 bonds here and there are 3 bonds, the 3 hydrogen bonds here. Now, if they say there is a 30% of A, that means like there will be also 30% of T, right? So if it is total, how much? 60. So how much is left from 100%? 40. The 40 will be again divided equally between the G and C. So now what they are asking for? They are asking for cytosine base. The answer would be what? It is 20. So this is how we can solve it. Next one. The concept of genes is not static but dynamic. Arrange the changing concept of a gene chronologically from the earliest to latest. Now they are asking you to arrange in a chronological order again. Now in this question, you should not again think about the years. You should not try to remember the years for all these events. So this is again one conceptual logical question. Just by applying your mind, you can answer it. See, what was the first thing that came into existence? You see, like if you just observe these two things, you clearly know that it is like first was metabolic block pathway that came. It was given by Garrod while experimenting with the alpha ketonuria, studying the alpha ketonuria, and next it was like one gene, one polypeptide, which was by Ingram, right? And next, if you know about the isolation, you know it like it was like much before when it was taken place. And the last thing that came into existence is like all after like first thing like they have called it as metabolic block, then you call it as like one gene, one enzyme, then they call it as like one gene, one polypeptide. These things refined, and at the end like we got a definition that it is a functional product, and functional product can be directly an enzyme, directly a protein, or it can be a RNA, anything, right? So this was the last. So that is very sure that A would be last, and the Frederick mixture it will be the first. So just try to arrange it, and you know it like it is first C and then came the D, like it is B, C, D, A. The correct answer to this question is B, C, D, the last one. Sometimes you have to not think about the years, you have to just apply your brain and the logic, and this will not come overnight. I will tell you again the same thing. If you practice the questions, if you see like many number of questions which are relevant to your syllabus, which are related to you, you try to improve your analyzing skills where you will not be directly knowing the answer. Also keep this in mind. Out of 120 questions, it's not that whatever questions you attend, you will be knowing it for 100% sure. Like there will be only like 50 to 60 questions will be direct, where you have studied directly, seeing the question you will answer it. Rest will be like analytical, less will be conceptual, where you have to apply your mind, think about it, and then try to answer. So this is how it will be. Do not be in the impression that in the exam I have studied well, I have completed the syllabus, I have done like 4 to 5 times of revision, so I will go and attend the exam. In the exam, out of 120, whatever like I know the answer, I will keep it and I will get a good rank. No. Keep it in mind, even if you are prepared well for this exam, only 60 to 70 questions will be direct and sure shot. Rest you have to think, you have to analyze, only then you will be able to answer it. Don't keep any predetermined mindset before going to the exam. Next one, it is regarding the TI plasmid. So TI plasmid is associated with the which disease? I have clearly explained in the classes of biotechnology, like we have discussed few things in biotechnology classes. I completely talked about rDNA technology. rDNA technology, which is the most important from the biotechnology for your ICAR PG exam for the plant sciences syllabus. So if you have not yet watched the video, the links will be available in the description box. Just go and watch the two videos I have posted. So that will be a lot more beneficial for your exam. And if you're a student of master course, I have also uploaded the notes. So just go through the notes once. So your revision will be done. Now coming back, TI plasmid, you know it. Like when it is TI plasmid, it is like crown gall disease, and it is from which pathogen, which organism, whether it is bacteria, whether it is like fungi, virus. Know it like it is bacteria, right? Okay, fine. Instead of crown gall, if they have given like bacteria, they have given like virus, they have given like fungi, they have given nematode. Even in this case, you should be able to answer it is like bacteria. Okay, bacterial disease, crown gall disease, Agrobacterium tumefaciens. Next one. So I have given bit detail about this TI plasmid and also like how it will cause the infection. Now one important thing to note here is like even in the exam, in the next year exam, if they ask like whether it will create the infection in the monocots or dicots, what you will answer? Is it both, or is it dicots, or it is monocot? So this is specifically for the dicots, not on the monocots, because they monocots do not have that receptive phenolic compounds which are crucial for this action of this particular pathogen. So that I have clearly indicated here. Just have one reading of it, so they will be able to answer this question and understand it. So in the exams, in the upcoming exams, if they are asking anything relevant to whether it is monocot, whether it is dicot, and why it is only dicot, why not monocot, and what is the mode of action of this, so you should be able to answer it. Next one. So which term is commonly used to describe the specific sequence of the bases recognized by the restriction enzymes? So the name itself suggests, like I told you in the enzymes for DNA manipulation in the biotechnology video also, like whenever they say that it is recognized by the restriction enzymes, itself means that these enzymes recognize certain sites in the DNA sequence recognize certain sites, they are called as restriction sites. Why it is called as restriction site, why the name is restrictions, everything I've clearly explained in the video, so please go and watch it once. So the answer to this question is like the restriction site or the recognition site. So this is the answer. Next one. Again, we came back to the assertion and reasoning time. So the assertion says that genetic code is degenerate, right? Like this is from the features of our genetic code. So if you do not have this features, should be on your tip of your tongue for sure. Like this is a must and should that you have to read, like features of genetic code. All the features should be in your tip of the tongue, so that anything that is asked in the exams you should be able to answer. The first thing it is saying like about the degenerate behavior of the genetic code. This is not the statement, this is the assertion and the reason, so you have to find it out. The reason says the genetic code is nearly universal. Now you have to just think like whether there is some connection between the assertion and reason or not, right? Like how to do it, like you have to decode the meaning. So what do you mean by degenerate? What do you mean by degeneracy? Like you have different terms, you might be knowing with this from the different terms. Degeneracy, the genetic code, it is like having the degeneracy. So what is this degeneracy? So like let us take one codon AUG. So it codes for methionine, right? So but methionine, if you know, like it is only coded by AUG, but what about the other amino acids? So we have several amino acids which is coded by more than one codon. That means the same amino acid, it is coded by the different codons, right? So this is what the degenerate behavior is. Like single amino acids can be coded by the more than one codons, and if like it is a one codon, the example is methionine. But the other amino acids, like how many number of amino acids we have 20 standard amino acids, and which are coded by how many codons? Like there are total of 61 codons which code for this 20 amino acids. Rest 3 are like stop codons which do not generally code for any of the amino acids, right? So these 61 codons, like if there are 61 for 20, that obviously means that each amino acid will be coded by more than one codon, right? So this is what the degenerate behavior tells us. Next, it is saying the genetic code is nearly universal. So why it is not completely universal, nearly universal? Because we know that the amino acid not always be coded from the codon. Sometimes a same codon can code for the different amino acids. Like if it is amino acid 1, like it is amino acid 2, the same codon. There are exceptional cases where the same codon will not only code for the specific amino acids but also for the other amino acids. Not for all, but for few exceptions. So that is why it is like not completely universal, it is nearly universal. But what is the connection between these two? There is no as such connection, direct connection. So the answer for this question will be like both A and R are true. They are the features of our genetic code, but the R is not the correct explanation of A. Okay. So now what to recall? You have to recall the features of the genetic code. It is like triplet codon, it is commaless, it is unambiguous, it is like non-overlapping, it is universal, like nearly universal, it has a polarity, that is direction, and it contains the start and stop codon, and stop codons like nonsense codons. What are these nonsense codons? Like if you remember, like it is like UAA, UGA and UAG, right? So these are the three different. It is like opal and UA is ochre, Greek terms, and this UGA is amber. Sorry, this is amber and this is opal and this is ochre, right? So these are the three different names for the stop codons, which is most important. Try to remember it. And also the features of our genetic code is very much important. The next one, dash is a second mutation that compensates the effects of the original first mutation. Choose the most appropriate options from the below mentioned options to fill the gap. Here there are two things which needs attention. One is compensatory and one is like suppressor mutations. So this is where like you will stuck. You will be like confused like what to answer, because in the question we have a keyword which is saying the compensates. So then it can be also the answer, but according to the definition we know like suppressor mutation is the one which will actually suit the above definition. So first like let us try to decode it. First one, what is forward mutation? The forward mutation is like going from the wild type like the mutant, so that is what we call it as the forward mutation, and the supplementary mutation, so there is not a standard term, you can skip it. Next, the suppressor mutation. So in the suppressor mutation, if there is a mutation 1 which has suppressed the wild type and induced the mutant phenotype is being induced in the individual. So there will be like second mutation which will actually suppress the mutant phenotype and it will give the similar phenotype as of wild, not the same phenotype, it will be like similar phenotype. That will be like suppressor mutation. And the what is compensatory mutations? So if you look at the compensatory, although like it is not available in our standard books that we read, but if you want to know the meaning, compensatory is just compensating, like how it compensate, like where the second mutation will try to actually compensate the effect of the mutations by changing the phenotype. It won't be like entirely mutant and it will be again not entirely the wild type, it will try to suppress the activity or the phenotype that is produced by the mutate. So that is what the compensatory mutation means. But according to this definition, like second mutation that compensates the effect of original first mutation, if this is asked according to the textbook definition, like you have to opt for suppressor mutation only, because in the books like the genetics books that we read, we have not find any term like explaining about the compensatory. So that's the reason. Although like if you find it in a Google, you just Google it, then you will find some meaning which is almost same as like suppressor mutants, but you have to go for the standard terms that has been defined. Okay, so here the correct answer is like suppressor mutation. And also like when we are talking about suppressor mutations, the mutation will not be on the same position. It will not be on the same position where the mutation 1 has taken. The mutation 2 will not be on a same position, on a same nucleotide stretch, on a same nucleotide base it has taken. Okay, but it will be on the same gene which will try to like revert the phenotype of the mutant. So that is what the suppressor is, like the effect of mutation is like reverted. Okay, so I try to give the explanation here, so you can just read the things here. So there is also one mutation like reversion mutation. The reversion is nothing but, here the reversion means like exactly the mutated DNA sequences changes back to its original wild type. When from the mutant, if it is getting exactly converted into wild type, such type of mutations it is called as reversion mutations. And in case of suppressor mutations, the two mutations actually coexist. If there is a M1, when M2 occurs within the certain gene, both will coexist, but the second will compensate the effect of the first one. So that is what it means. And if you talk about the compensatory mutations, they do not directly revert to the original mutations as I told you, but they occur at the different site in a genome, like often like they will be called as like same gene, but it's not very clear. So still I have tried to put the meaning here so that you will not have any confusion on this, but answer to this question is like exactly matching with the suppressor mutations. Okay, so this is the for this one. Next we have like during which phase of a cell cycle the chromosomes most condensed and visible under the microscope. So now they are asking about the different phases and asking like which will be the most condensed and visible, two things here. So we know like it is metaphase, and again if you know it, whether it is like meiotic metaphase or the mitotic metaphase, what you will answer? Whether the meiotic metaphase or the mitotic metaphase will be like most condensed. What you answer for this question? So you have to know this, like whether it is both or it is the one, and if it is one, like why it is the one, why the other one will not have the most condensed and the visible. So answer here, it is like metaphase. The mitotic metaphase, it is like most condensed and visible, which is like actually used for the studying of the chromosomes for the karyotype analysis. Chromosomes. Okay, now coming back, like now they have not directly given you the stage, like instead they have given you as like G1 S phase and G2 phase, plus it is like after that M phase. Now here like they have intentionally mentioned the mitosis so that you should be knowing that this is the answer, that in this phase the chromosomes are most condensed and the visible under the microscope. So now from the starting chapters of genetics, from the mitosis and the meiosis, you should be like, you will be always learning it from 11th and 12th. But what becomes important, you have to not neglect it. You have to strictly revise the things, know the things like what are the different events that is taking place in the mitosis, what are the events that is taking place in the meiosis, the substages of the mitosis, the meiosis, again the substages of the meiosis 1, the meiosis 2, and again in the meiosis 1 we have like prophase. In the prophase we have again 5 different stages. All of this, not only the names, but the what kind of changes that is occurring in a cell, within a cell. So that things are also very important. Please make sure that you know it. And I also mentioned like why the mitotic metaphase is suitable, not the meiotic. You know, like in the meiosis there is an event of like crossing over, the recombination that is taking place, which will not, and this is in the prophase, you know, like in the prophase there will be like genetic recombination, and after prophase like there is a metaphase. So what kind of events that will be taking place in the metaphase that will disturb the study of my chromosomes? Like you know about the chiasma, right? So that is like visible at this stage. So because of which there is no possibility that we can study the chromosomes correctly. Okay, so that is why like it is always the mitotic metaphase, to in a simple words, that why we do it only in this phase. Okay. Next thing. So now it is about the match the following. Okay. Now they have given the list of scientists and their discoveries. So first thing, T.H. Morgan, we are well aware of this, and Kelvin Bridges, we are familiar with Oliver, and we are also familiar with the Benzer. So let's say like how it is. As soon as you see the chromosomal theory of inheritance, we can mark it as like T.H. Morgan, and as soon as we see the chromosomal non-distinction, it is for the Bridges. And Benzer, like he gave the deletion mapping, and Oliver, it is like divisibility of gene, or they can also give as like intragenic recombination, and this was in for which gene? Lozenze gene in case of Drosophila, for which it was discovered, right? Although like the credit is given to Sutton and Bovary, but the clear-cut evidence for the chromosomal theory of inheritance or the heredity was provided by the T.H. Morgan, right? So that you have to remember. So answer to this question, just mark it, just know it where it is. Next one, the chemicals that store the energy for the majority of work within the cells are called as. Now these things might confuse you people, because the energy source, the energy currency, what we know from the cells, it is like ATP, but we have to know like how this ATP comes. So this ATP you have to know and try to link it with these biomolecules. First, what are lipids? Lipids are like long term energy storage, right? So these are long term energy storage, and the proteins, you know, they are involved in like transport, they are involved in metabolism, they are involved in regulation. So this is its activity. And when it's come to the carbohydrate, it is like short term energy storage, again short term storage, and it's enzymes, so it has the activity of the catalytic activity. So this is clear. Now if you know it, like from the carbohydrates, like the glucose, the glucose is actually metabolized to produce the ATP. Considering this, the answer to this question will be the carbohydrate here. But as soon as we see like energy currency or the energy, like we always feel that okay, it's ATP. So you have to try to link it from the ATP to glucose and the carbohydrates. That's how you'll get the answer. So this is how you have to like train yourself for such questions where you directly do not find the answer from the options, but you have to try to link the question to the options by somehow. Next one, list 1 and 2. Nullisomic, monosomic, they are trying to ask you the formulas. This is very simple. See, I'll just list out the formulas here. You can match it by yourself. Nullisomic, it is like 2n minus 2, and whenever I talk about the monosomic, it is like 2n minus 1, loss of one chromosome. And when I talk about tetrasomic, so what is tetrasomic? The addition of a pair. And when it is like trisomic, the addition of single chromosome. And when I talk about the double monosomics, and it is like 2n minus 1 minus 1. So based on this, you can match it by yourself. Double monosomic is done, nullisomic is done, the monosomic is done, trisomic is done. All these things are done. Next, the human beings have the large size proteome. The principal and more significant reason for this is, so you might have not seen such kind of things in the books that you refer and the chapter you refer from the BDC if you are following. So as soon as you see it, like don't be scared. So just try to like break it. So what is proteome? It is like complete set of proteins that are present in a cell or organism. Complete set of proteins that are present in the organisms. So they are asking like human beings have a large size proteome, like the complete set is very large. The principal and more significant reason for this is humans have extremely large genome. This is not entirely true. Like there are a lot of there are few organisms which have more genome size than of humans, right? So this is not true. And the individual gene of the human genome may encode many different polypeptides. Okay, it seems relevant. Next, the human beings are the most complex. No, like there are also like the other organisms which are complex, and that is not related to the question. Yes, even though like human beings are complex, but it is not related to the question they are asking regarding the proteome. And last one, the human genome contains the millions of different genes. So millions of different genes is fine, but this is not exactly trying to answer the question which is asked, right? Like how many millions of genes it has? Like is it true that it has in the millions? No, right? Like the question, how many like genes we have on an average? We have around 25,000 genes, not in millions at least. 25k, 25,000. Okay, I missed this term, millions. Okay, this is wrong, not millions. We have approx like 25,000 genes that are present in the humans, in the human genome. So now according to this, the second one is like the correct option for this, since the individual same genome, because of a mechanism called as alternate splicing, the same gene can produce like more than one type of enzyme or the more than one type of polypeptide. If the same gene is able to give the more than one type of protein, one type of polypeptide, which is again going as protein, that means like we'll get large size even with the limited number of genes, right? So because of this, like second option is right. Okay, so proteome, what is it? Proteome, I given you the definition. Next, the longest phase in the eukaryotic cell cycle. If you just Google it, you'll get a lot of different answers. But one thing like you can clearly say here, out of G1, yes, G2 and the M phase, you know this is the shortest one. That's for sure, right? Like the M phase is the shortest one. The interphase that you have, this three belongs to the interphase. So this is the longest phase. But again, in this interphase, which is the longest phase? So before that, let's see like what happens in the G1. Like it is like mostly the protein synthesis will happen and it will prepare itself for the division, and G2 also most of the protein synthesis, and in the S phase there will be like actual DNA replication that take place in this case, and in the mitosis or the meiosis it will be like dividing phase, right? And also like some 0.3% of DNA replication will also occur during the meiosis. Fine. So this is what we know. But regarding the duration, so if you want to know the duration, the G1 is quite variable. So the range of duration in the G1, it is like from 6 to 12 hours. Like the range is there is a wide range from the for the different organisms. It is not uniform. It ranges from 6 to 12. But when you talk about S phase, it is like 6 to 8 hours. Maximum is 8. And for G2, it is like minimum 3 to 4 hours. So if you want to summarize, like it is G1, like S, and the G2 generally. And the longest is like obviously the G1 here. And the smallest, if you want to, like here it is after the M phase, whether it is meiosis or the mitotic phase. Okay. So based on this, what will be the answer? The answer will be like G1 phase. Next one. So dash can occur in either the first or second meiotic division to produce the abnormal gametes. Choose the correct answer. Again, so this is again confusing. So all of these things will lead to the abnormality, but they are asking whether it is in the first or second meiotic division. So here, these are all the indirect effect, what you are seeing. But the third one, the non-disjunction, which is a mechanism where the chromosomes fail to separate. Chromosome fail to separate during the cell division, because of which we'll get the abnormal gametes. So this was non-disjunction. C.B. Kelvin Bridges, right? We talked earlier. So he gave this concept or explained this one, non-disjunction behavior. So the answer to this question is non-disjunctions. That is how like sometimes you have to think and you have to choose the most relevant one in the options. That's how you can do it. Next thing, like unlike Mendel's experiment, the Bateson and the Punnett's experiment with the speed P demonstrated that in the F2 generation, the genes for the flower color and the pollen length. So even before looking at the options, you can say that when these two like experimented, they did not get the Mendel's same as the Mendel's ratio. The ratio was in the test cross was like 1:1:1:1 test cross ratio, but these two got the ratio which was like 7:1:1:7, which was significantly different from the Mendel, by which they came up with the concept of the linkage, right? That means the genes do not assort independently. So this is like do not assort independently, because the genes for these two different traits or the characters, they did not assort. So now I was using like the word character and a trait. This is again most confusing between the many students. What is the difference between the trait and the character? I want you to comment on this video. Just comment on this video with the answers. I'll just look at the answers. I want to look it. I will be loving to see your response and the engagement that you show to our platform. Okay, so this is about the answer. The answer to this question is like do not assert independently. Next, the process of turning on the expression of the genes due to the environmental condition is called as. So what is it called as? Turning on. They are talking about on. If they are talking about off, so it is like repression. If they are talking about the on, like induction. I agree, like you might have not really read this anywhere in the books that you refer for the ICRPC exam, but just by applying the common sense, you can say that repression is something when it is like switched off, and the induction is when something is switched on. And when we talk about constitutive expression, irrespective of the environmental conditions or the regulatory mechanisms that we have, if you remember something from the Lac-Operon concept and the gene regulation, irrespective of whatever might be the environmental conditions, whatever might be the regulatory mechanisms that are taking place, it will be always on. So that is what the constitutive expression means. The expression will continue. And when it is like enzymatic catabolism, it is like breaking down of a compound with the enzymatic process. That is what it is saying. But these are not related to the question. The only things that are related is to, and repression is off and the induction is on. So this is how you can answer this question. Okay, the next one, again the statement questions. The given below are the two statements. The one is saying like amino acid is in the protein synthesis is methionine in eubacteria. So in the eubacteria, the first amino acids, what we call it as start codon. Start codon is AUG and it codes for the methionine. So this is what we generally know, right? And they are saying that this is in case of eubacteria, it is methionine. And we also know that it is like formal methionine. And formal methionine in which organisms and the methionine in which organisms, this is what we have to figure out. And the statement 2 says that the first amino acids in the protein synthesis is like N-formyl methionine in case of archaea. Now remember, like formal methionine, it is like specific to the prokaryotes, that is in bacteria, and methionine, it is in case of both archaea and the eukarya, both the domains. And for the bacteria, so it is the formal methionine. If you remember, the start codon AUG, generally it codes for methionine, right? It codes for this amino acid methionine. But during the initiation of this translation, so this codes for the formal methionine in case of what? In case of bacteria. And after the start, in between, if the same AUG comes, again it will come back and produce the methionine even in the bacteria. Also this you have to remember, only during the starting it will produce the formal methionine, and in the later also, even in the bacteria it is methionine. And when it comes to the eukarya and bacteria, remember it is not the formal methionine, it is the same the methionine, which is like start codon, and also same will be for all other in between also, that will be like methionine. The AUG will always code for methionine. So according to this, both the statements are right. Here it should be like methionine, it should be like archaea or eukarya, not the eubacteria. Here it should be like the eubacteria or the bacteria, right? So this is like both the statements are wrong. Next one, the number of possible combinations in a polypeptide containing any 7 amino acids is given by. Again, you see some maths, like you just be scared. Okay, I have not prepared for this, like I have not seen anything like this before in the books, like how should I answer it? So again, it's very simple to answer. Just try to decode it what they are trying to ask. The number of possible combinations. It's simple mathematics. Like, see, they are saying like there are 7 places. So there are 7 places, and they are saying that you have to arrange any amino acids in the 7 places and tell us like how many combinations you can make. So how many amino acids are present total? So out of 20, we can place each one of them in these blanks and try see like how many different combinations we can make. See, in the first place, how many amino acids we can keep? We can keep 20. In the second place, how many we can keep? Any of them in out of 20. In the third place also any of them out of 20, and the fourth, fifth and sixth and seventh, all of them, any of them from the 20. So is it like 20 power 7, or the simple formula for this is like n power r, right? So this is the formula. So if you apply it, it is like 20 power 7. Sometimes students get confused like whether it is like 7 power 20 or 20 power 7. So now know it like total number of whatever is like whatever is available, the sample space, it will be like in this area, which is n, and how many it should be having, like 7, it will be like equal to r. So that is how you can remember. And same thing, you know that there are 3 places. This is in case of codon. And how many nucleotides you have? You have 4 nucleotides. How do you arrange this? What are the different number of combinations that are possible here, if you remember? So again, 4 power, it will be like 3 places 3. Like here you can place any of the 4, here you can place any of the 4, here also you can place any of the 4. It will be like 4 power 3. How much it is? Like 64. We know it, right? There are 64 codons which are coded by like 4 nucleotides bases. So the answer is like 2 power 7. So these things, like you try to like see, there are not many different kind of questions that can be asked in the mathematics, like related to the mathematics. So there will be only limited number. If you just solve all the different patterns of such kind of questions, anyhow, like you will be easily able to answer without any doubt. Okay, you need not to prepare for it. Again, the one question from the population genetics. I have clearly explained the population genetics in the YouTube channel. So the links I will be also making it available in the description box. Like if you have not yet watched it, if you are not a student of our master course, then go and watch it in the YouTube. And if you are a student of master course, I believe like you have not missed this in this course. Like I have already explained in detail about this R.D. Weinberg's law in the population genetics. Okay, now let's get back to the question. So in the population of R.D. Weinberg equilibrium, if the frequency of homozygous genotype is 0.64, it is like 0.64, then what will be the frequency of dominant allele? So first thing what they are saying that the population is in R.D. Weinberg equilibrium. So now like in order to answer this, you have to know like what is R.D. Weinberg equilibrium, what does it exactly mean, and when it is in the R.D. Weinberg equilibrium, what kind of derivation we have, that is like 2pq, like p square plus 2pq plus q square is equal to 1. Why it is equal to 1, how this formula was derived, these basic things will be much more needed and crucial to answer the questions. Anyhow, like you can answer this very easily, but day by day, like the competition rises, the kind of questions will be twisted, they won't be always direct. So knowing the concept, knowing the right concept is always important. So please know each and everything about the basic things, not in detail, not in depth, the basics about the R.D. Weinberg's law. Okay, now starting with like when it was proposed, whether it was independently or it was like together. Just comment on the video like whether it was like independently discovered or came up with this equilibrium, like this particular equation, or it was like together, and also with the years. I want it in the comments. Now they are saying that homozygous dominant, can I say that if it is in the equilibrium, can I write it as like p square is equal to p into p, where p square is actually the frequency of the genotype capital A, and the p is actually the frequency of the allele capital A, right? So if it is in the equilibrium, I can write it as like p square will be obviously equal to the product of the allelic frequency. Now here if you just substitute p square is equal to p into p, now p square, so what is the value of p square? It is 0.64, and now p into p is p square. So now we want the value of p, which is also the frequency of dominant allele they are asking. So now it will be like root of 0.64. So what will be the answer? It will be like 0.8. And also like if P is equal to 0.8, then what will be the value of Q? Q will be like 1 minus 0.8, it will be 0.2. And what will be the value of 2PQ? 2PQ will be 2 into 0.8 into 0.2. 16 twos are, it is like 0.32, sorry, 0.32. Fine. So here the answer to this question is like 8.4. It is a frequency of capital A. And if I ask you about the frequency of small a, it is like 0.2. And 32, it is like frequency of heterozygous genotype. Frequency of PQ. Okay. Next one. So again, another match the following. So they are asking about the operon. See, operon thing has come like multiple times in the 2024 paper. So that's why I always say in the molecular genetics. So how you have to start your preparation. See, a lot of people like are worried too much about the molecular genetics, but for your ICRPG exams, there is like very limited that they will ask. See, starting you have to start with genetic material. You have to know about all the events, the scientists, like how the genetic material is discovered and the structure. The second thing will be structure of genetic material, not only the DNA, also with DNA go with the RNA and try to differentiate between these two. Next, after structure, like organization, what is the structural organization of genetic material. Next, you have to go for replication. You have to know the replication for both the prokaryotes and the eukaryotes. Next, the transcription. And once you complete the transcription, it is about translation. And again, it is for prokaryotes and eukaryotes. Translation, prokaryotes and the eukaryotes. And next, the regulation in case of prokaryotes and the eukaryotes, and what is the difference. And in the prokaryotic regulations, you have to particularly concentrate on the lac operon and the tryptophan operon. So that will entirely finishes it. And plus, like you can also read for the transposable elements. That will almost finishes most of the things from the book. You need not to follow the BDC page by page for the molecular genetics, which I have already explained in the genetic made easy video. And again, if you have not yet watched the genetic made easy video, the links will be available in the description box. You can go and watch it. Okay. And if you have skipped it, so that will be a serious problem for you. Do not skip the video, because I have explained in detail about like what you have to read and what you have to skip. So you have to follow it if you want to get a better rank. And if you are worried, already if you are doing well, you are confident about the subject, you know much more things than what I say here, then do your preparation, not a problem. But if you are worried, if you don't have a clarity, you want some guidance, then these videos will definitely help you. Coming back, there are promoter, operator, structural gene and regulatory gene. The promoter, so what is the function of the promoter? The promoter is like binding site for the RNA polymerase. I told you already in the previous slides. The operator, it is like site for the repressor binding site. The structural gene, it produces the functional gene proteins, and the regulatory gene produces the repressor protein. So just match it accordingly. So this is the answer. It's very easy. In the humans, dash trait are much more easier to identify than dash traits. Choose the correct words from the options given below by filling the blanks. So this is the one which actually might confuse you, right? And in the books also, they have not given the direct answer for this. So now this is your homework, like you have to find the answer for this question. And also I want some clear cut references or the source from where you are saying this. If something is answer, like why you are saying is, how you are saying it. I want it in the comment section. So I'll be reading the comments after like 24 to 48 hours. I'll come up with the answers and I'll drop the answer in the chat box, and I will also pin it. Not issue. Okay. So I want you to answer. Just remember the question number. It is like 30. Next one. Next, again, the one more match the following. See, whenever you see the match the following kind of questions in the exams, just take it as a boon. It's a boon for you. You can easily score 4 marks attending this match the following. First one is Streptococcus pneumonia, Neurospora crassa, bacteriophage, and TMV. And they are trying to link it with like in which of these like used for the establishing certain principles. Now, TMV, you know that it is like used for the RNA as genetic material. And who gave this? Who discovered that RNA is a genetic material? So it was by Frankel, Conrad, and Singer, right? So remember this. And the bacteriophage, T4 bacteriophage, why it was used? So this was used, if you remember, for the fine structure analysis that was made by the Benzer, right? And the Streptococcus pneumonia, and this was used for what? For the DNA is the genetic material, starting with the Griffith experiment. So this was used. The last one which is left, the Neurospora crassa. And this is used for like one gene, one enzyme hypothesis, which was given by Beadle and Taunt. Okay, remember all of these things, try to link it and try to answer this. See, even if you just make 2 right now, rest of the options will automatically match it by you themselves. No need to worry about it. Next one. So, which of the following traits are observed in the eukaryotes but not in the prokaryotes? Okay. So, this seems simple unless you get this term phagocytosis, which you might have not heard directly. So, now I told you, I always tell you in this prokaryotes and eukaryotes, in this chapter cell ultrastructure, you have to make a clear cut difference between the prokaryotic structures and eukaryotic structure, which are specific to prokaryotes, which are specific to eukaryotes, and which are common to both. Right? If you make this, only you will be able to answer it. See, even if you know that 70S ribosomes, it is both in prokaryotes and eukaryotes. Prokaryotes, it is present in the eukaryotes, it is present in the this ribosome, it is present in the mitochondria and like chloroplast. You know it, so you will be knowing this. And nuclear division, it is like specific to eukaryotes, and binary fission, it is specific to, it is both common in like eukaryotes and prokaryotes. Why? Like prokaryotes, the main division is a binary fission, but in the eukaryotes, for the mitochondria and the chloroplast, it is again the binary fission. And the presence of nucleolus, it is specific to eukaryotes. But based on that, they are asking like following traits are only specific to eukaryotes. You know it is like E and you know it is like B. So these two options are there. But what about C? Whether it is in the prokaryotes or not. So phagocytosis, and there are two important terms, phagocytosis and the pinocytosis. Remember, this is like cell eating. Simple way to explain this: cell eating, and it is like cell drinking. And this is limited to the eukaryotes, not found in the prokaryotes. You have to remember this. There is no other thing here. So now based on this, the answer would be like B, C and E only, not this one. Okay, these areas, like some might commit mistakes because not everyone will be sure whether that the phagocytosis, pinocytosis is common to both or is specific to one. That is why, like, while you prepare, so if there are two different things, like if you have like categories of prokaryotes, eukaryotes, whatever mechanism, whatever things you study, try to compare. So see like whether it is in prokaryotes or eukaryotes, or whether it is common for both. So this way you have to reduce it and prepare, so that if this kind of questions is asked in the exams, you will be able to answer it. Okay, the next question is like, which of the following organisms naturally possesses the CRISPR-Cas system? Okay, so they are asking about the CRISPR-Cas. So what is the full form of the CRISPR-Cas? Clustered, regularly interspaced palindromic repeats. And this was once asked in the PhD paper also. The full form, they are asked the abbreviation, like this abbreviation full expansion was asked, where the options were, one was like interspaced and other was like interspersed, and there are also other two options. So this might confuse a lot of you, right? Like even it will be confusing for me if I have not specifically paid attention to this word, right? So that is why, like, whatever abbreviations you have, like the expansions, like you have ISTA, you have several other things, you should be able to like know the exact expansion of it with spelling by spelling. So if you know it, that will definitely help you. So this was for CRISPR-Cas. They are asking like this system is available in. So to just let you know about the history, like these are like categories, or like these are under the category of site directed nuclease, which will make the specific cut in the DNA, in the genetic material. And again, in the SDNs, we have 4 different types based on the order of the discovery. They can be like mega nucleases, and they can be like zinc finger nucleases, talents based on the discoveries, zinc finger nucleases, and lastly, like most widely used in current generation is CRISPR-Cas system. And they are asking about this CRISPR-Cas. So the CRISPR-Cas, this system is available, which is actually a natural immune system which is present in both bacteria and the archaea. And in the example, the Streptococcus pyogenes, so this is one the commercially used for the studies, like for the research studies, like the CRISPR-Cas is the one, like in this, like Streptococcus pyogenes has been widely used. So it is under bacteria, but also remember, so here anyhow the answer is bacteria, but if they also give the archaea will be also the answer. And I've given the full form of the talents. What is talents? Transcriptions, activators like effectors. So few details about this is also present here, where have tried to give you the differences in between the like meganucleases, the zinc finger and the talents. And the last, it is like currently used, mostly used one, CRISPR-Cas. Now I told you about the order. Like order is same. Like I think like I've okay, it is talents. Okay, next zinc finger. No, it is actually zinc finger. First is like meganucleases. Second one is zinc finger. Next the talents, and the third one is like CRISPR-Cas system. Okay. So that's why they have mentioned the first generation, second generation, third generation, and the fourth generation. So try to read the things from this slide. So if it is visible, I don't think like whether, I don't know like whether it is visible or not. So if it is not visible, anyhow, like you will be getting the PDF. Yes, I will be sharing the complete PDF like what I am discussing it right now. And to get the PDF, I already told you like you just have to message on this number. Just drop a message. There is a quick activity for you. Once you complete it, you will get this PDF for sure. Okay, so that completes. So this completes the complete set of questions that came from the GPB for the 2024 subjects. Okay, I hope like this particular video is informative. It takes a lot of time and efforts to prepare the content. See, explanation is different thing. Like I can do it in just 1 hour or half an hour, but preparing this content, getting in the right format, getting in the PDFs, getting in the PNG form, like getting into the PDF to provide it to you and getting all the relevant informations, that's a very time consuming task. And if you feel that we are making some dedicated efforts for you people, because we also have our exams, we also have our research, we also have our like ARS exam mainly, but still we are trying to make our time because this is a crucial time. So we have supported you till the year end, till the day, and we want to do it till the exam day. Okay, and there are almost like 800 plus students who have registered for online master course. It is really going well, and we have a lot of positive feedbacks about the course. Like if you have not yet registered and wanting to enroll, it is not compulsory for everyone. If you are preparing well, you are confident about yourself, just enrolling the mock test bundle will help you, right? But mock test bundle is compulsory. No matter like from whichever platform you take, you have to take mock test bundle, and also make sure that whatever questions that you answer, that should be of some standard. That doesn't mean that you answer any kind of questions. Wherever you choose to attend the whichever platform you choose to attend the mock test, make sure that to analyze who is preparing the mock test, whether they are qualified or not, whether they have cleared this exam or not, whether these questions is like common to all other agriculture, they are simply putting it in the ICRPG and just giving it to the students, or they are seriously dedicated to solve all your queries, solve all your doubts. Based on that, try to get the best mock test bundle, and we also provide the mock test bundle, which will definitely help in your preparation. We have topic wise, subject and whole syllabus that you can enroll, and links are available in the description box. And I have one important activity for you people. So now at this stage, if you think like you want the other series, like you want the part 2 of it, the plant pathology, the microbiology, the seed technology, the 2023-2022 paper, and the most importantly the revision series, if you are waiting for the revision series, I want at least 500 plus comments on this video. And you know, like the number of students in the master course is more than that, but still I want at least 500 engagement, and I want to see like whether you people are actually getting benefited or not. So if you are getting benefited from the videos we are making, I want you to comment on this video that you are getting benefited, and or you can just mention that we need the revision series, rapid revision series. Just comment that we need rapid revision series. So we will prepare the complete PPTs for that, and we'll compile all the genetics in one video, all the microbiology in one video, all the seed science in one video, and complete plant breeding in one video. So once you watch this 2 to 3 hour session, like it will be like obviously 2 to 3 hour marathon sessions, then most of the things will be very clear, and you can go and write this exam very confidently. And I'm pretty much excited to come up with this revision series very soon. So I'll be waiting for your comments, I'll be waiting for your likes, I'll be waiting for the love that you show to our platform. And if you like our content, please like, comment and share and subscribe our YouTube channel AgriRate. Thank you so much for watching. See you all in the next video.